Welcome. Thank you all for coming today. My name is Lynn Bonnet, and I am president of the Greater New Haven Green Fund. We thank Southern Connecticut State University's Department of Public Health for partnering with us and hosting this event in this lovely new community room. It is customary to acknowledge the indigenous cultures, the Quinnipiacs, that lived here prior to the arrival of the colonists. Now, we can also refer to this land as being part of the Quinnipiac bioregion. The Greater New Haven Green Fund is a nonprofit public charity that awards competitive grants for small grassroots organizations that promote environmental quality, public health, and equity in the communities in the Greater New Haven region. Since 2011, we have awarded over $800,000 in the form of 200 grants and sponsorships that engage and empower citizens and organizations to help create clean, healthy, and environmentally sustainable communities. One of our grants in 2017-18 funded a project at Southern to help decrease the use of single-use plastic water bottles on campus. Rebecca Pajinski, who was a graduate student in the Department of Public Health, collaborated with Southern's Office of Sustainability to study the barriers to using their reusable beverage containers program and the use of the hydration stations. The Office of Sustainability helped recruit participants for the study. Rebecca did educational outreach and held blind tasting tests to compare the taste of commercially bottled water to that of the hydration stations. She promoted the use of the hydration stations. She offered a small financial incentive for those that used the reusable beverage container program. After 6 months, she surveyed the participants about whether they used the water filling stations and reusable containers. The final results were that 75% reported using the hydration stations, while 13% reported using the reusable beverage container program. While 75% thought that the water from the filling stations was safer, about 50% bought water on campus because they forgot their reusable bottles at home. For conclusion, quote, more research is needed on how to reduce single-use plastic water bottles on campus. Furthermore, 34% of the participants think recycling single-use plastic water bottles completely eliminates the effect of plastics on the environment, which calls for more education on the recycling process. And granted, this study was done in 2017-18, so almost 10 years ago. A few years before this project and the installation of the hydration stations around campus, Dr. Vincent Breslin was investigating the presence of microplastics in Long Island Sound. We will hear from him a little bit later. We now know that plastics are everywhere, including in our bodies. In 1950, 2 million tons of plastics were produced. After 2000, the pace accelerated. We are now producing around 400 million tons worldwide per year. Current predictions are that we will double this amount by 2050. I won't be here then, but many of you will be, and your children surely will be. We need solutions. I'm very much looking forward to learning from today's speakers. I hope that you enjoy the talks and find them useful. At the end, we invite you to share food and join in the discussion. We want to hear from you what you think, what stood out to you, and your ideas for how to move forward. Also, we have some wonderful student posters, presentations on their current work in this field. I hope you visit and engage with us. So let's get going. I'm going to introduce our next speaker. Dr. Annalena Njoku is a full professor in the Department of Public Health at Southern. Her academic and research trainings have focused primarily on community-based health promotion and disease prevention efforts at the local, regional, and international levels. She earned her Bachelor of Science in Public Health from Rutgers University, Master of Public Health from Boston University, and Doctor of Public Health from Drexel University. Her broad research interests include social determinants of health, health disparities, cultural competency in higher education, environmental health equity, and rural health, and the scholarship of teaching and learning. She currently is the chairperson of Health Equity and Anti-Racism Peer Committee at the Department of Public Health at Southern, and has graciously agreed to partner with us to make this presentation today. So please welcome Dr. Amelia Mitchell.
Really excited about this event because we are bringing together community members. There's students in attendance from various institutions, both within Southern, University of New Haven, and area universities. And we also would like to provide insight into the effect of plastics on our health and our human environment. And I also just want to quickly acknowledge members of our department. We have our in the back. He has helped to provide a lot of support. We have some faculty members, students, alumni, public health alumni that are going to discuss this issue of microplastics and the effect of human health. And I also want to acknowledge that we had a microplastics and human health subcommittee of two Master of Public Health students, which I really want to acknowledge, Taryn Pierce in the back, as well as Tani Cookson Mohammed. They were really great at just helping to get this event together. So we are looking forward to hearing from the presentations from our speakers to provide insight into this really important public health issue. Then afterwards, we are going to have some networking, and we have some really great student posters as well that are going to highlight this issue. So we thank you for your attendance and taking the time to spend with us today, and we hope that you enjoyed this event today. Thank you so much.
Our next speaker is Dr. Jason White. Dr. Jason C. White is the director of Connecticut Agricultural Experiment Station. He received his PhD from Cornell University in environmental toxicology in 1997. After a one-year postdoctoral position at the agricultural station, he joined as a staff scientist in 1998 and has been working on issues related to food and environmental safety ever since. He is a member of the Connecticut Academy of Science and Engineering, is a commissioned official of the U.S. FDA, and has faculty appointments at Yale University, the University of Connecticut, and the University of Massachusetts. Please welcome Dr. White.
Funny story, as noted. There we go. I got my PhD at Cornell University, but actually in 1992, I came and interviewed for graduate school with Professor Gabe Benoit when we were both much younger and had different color hair. But unfortunately, he didn't have any funding available at the time, so I had to go to Cornell. But we did okay. So I would like to cover some of the work that we've been doing. See if I can get my pointer here. On micro and nanoplastics. Yep, there we go. At the Ag Experiment Station. But before I do that, and this is really directed at the students of the room, I did want to talk a little bit about what the Connecticut Ag Experiment Station is. So every state in the US has an agricultural experiment station. The one in Connecticut was actually the first one that was founded in 1875. So for those doing the math, that's 151 years. So there's a couple of things that make us unique. The first of which is the original state law that was written in 1875 actually said we had to be independent. So we are a government agency, state government agency, and you can see our funding breakdown there. But why that's unique is because every ag experiment station in the country after us, the decision was made to put them on the land-grant university campus. So California was second, that's at UC Davis. UMass Amherst was third. Every other ag experiment station, there's about 14,000 scientists working for the ag experiment station. All but our 110 or so are actually university staff and faculty and professors. So that means our mission's a little bit different because fundamentally we do research. We're not on a university campus. We don't do a lot of education and teaching. So that gives us a bit of a different focus. The other thing is that we're actually governed by what's called a board of control, which is also goes way back to that original law. So that means even though I'm an agency head, I am not a political appointee. The governor serves on that board. He or she is the president, he currently. But there are several other members on that board. So again, that gives us a slightly different flavor of what we do. Obviously agriculture is one of our core programs, but what's happened over the years is we've had kind of mission creep, right? Our agency motto from the beginning has been putting science to work for society, and what society needs has changed over time. So we now have large programs on the environment, which we're going to talk about today, food safety and public health, and obviously those are not four completely independent silos. There's a lot of overlap. So about 85% of what we do is research, but we are a state agency, so we do have some regulatory responsibilities. I just listed a couple of them there. The state beekeeper works for us. We have an inspection team that goes to nurseries, inspecting all material coming into and out of the state. I'm the state chemist. Again, it gives us a kind of a different mission and a different flavor. You can see our departments there. About 105 to 110 staff. The number bounces around a bit depending on the number of grants we get and postdocs we hire. We are also able to host international visitors where we're able to issue J visas. So we got about 15 people who have their own funding who are now doing research with us from around the world. We've got 2 postdocs from Brazil, somebody from the Chinese Academy of Sciences, somebody from New Zealand, so forth and so on. So just kind of give you a flavor of who we are. So to kind of set the stage, I think, some perspective on plastic production and use, I think is in order. You've already heard some of these statistics from Lynn, but and you'll probably hear them again. The one I like to kind of think about is, I don't know, for those of you who've seen The Graduate, which came out in 1967, there's this scene where Dustin Hoffman gets pulled aside and told about the future of plastics. But kind of more historical perspective, the first actual plastic was made in 1907. There wasn't a whole lot of use after that. It found its way into some of these products, but where things really started to take off is as we approached World War II and afterwards, as many things with the chemical industry just kind of took off. But DuPont got into the picture with the invention of nylon in 1938. But after, in that post-World War II period is really where we saw, this integration into consumer products at a large scale. And as we heard, between that 1980 to 2000 period, 2020 period, there was just tremendous use. Depending on whose values you're using and how you're calculating them, we're looking at, somewhere near a quadrupling of plastics production. As noted, we're at about 450 million metric tons right now. I pulled this data from OECD report in 2022, but I checked it this morning, and the 2024 value is about the same. In terms of recycling, secondary production, we're actually up to 29.1 million metric tons, which sounds like a good number, and it is a good number, but that's only about 6% of production, and it hasn't really been going up much in the last 10 years or so. So I work in an ag experiment station, so obviously our focus is on ag and food. So what about plastics and agriculture? Again, I just kind of threw up some key highlights. This scientist, Emmert, he was actually at an ag experiment station. He was an extension worker at the extension researcher at the University of Kentucky. And he's the one that's kind of credited with really first talking about and investigating how you could use plastics to increase agricultural production. His work kind of led to more greenhouse production or hoop houses or plastic siding, however you want to think about it. But basically trying to isolate an environment out in the field where agricultural production could be increased. Plastic mulch started in the 1950s, and obviously in New England, we see a lot of plastic mulch production, including at our farms where we do agricultural research, unrelated to plastics. You see it, agriculture is different. If you go to a soybean field in, Minnesota, you're not going to see much mulch production, but with specialty crops, it tends to be pretty heavy. So that really took off through the 1990s. The benefits of this are significant. I mean, it really minimizes weed growth. It helps you retain soil moisture. It helps you control temperature. And all of those, they save a farmer money. They increase yield. So there's some significant benefits to this, which is why it took off. 2019 was the most recent value I could find, where plastic production specifically for agriculture was at about 6.7 million tons. That's a non-metric ton. So that's about 2% of global production, which is, it's quite a bit if you think about it. So there's actually this whole field now that's called plasticulture, which is research and development on how to use plastics safely, sustainably to increase food production. And where you're seeing it, the most growth in the last 10 years are places like Spain, India, and China. That's probably not too surprising because those are places, among others, where the stresses on agriculture are going to be more significant, whether they're climate stresses or stresses in terms of the need for agricultural intensification, produce more food per unit area. So I should mention that this is just the production end, I have a little bit of work that we do on the food packaging end. I'm not going to talk about that at all today, but obviously when you talk about plastics and food and agriculture, that back end, that food packaging end is really significant as well in terms of amount used and in terms of the exposure it creates for humans. So why are we here? Let's talk about the toxicity concerns over exposure to plastics. So as noted, my PhD is in environmental toxicology. And plastics are an interesting case for environmental toxicology because, back to my days in graduate school and up until even relatively recently, where we as not just environmental toxicologists, really even toxicologists, where we tend to focus our exposure on those from those chemicals that tend to be toxic at really, really low doses. I mean, that's just historically where we've been. So, think about DDT, PCBs, dioxin, more recently PFAS. I mean, the EPA drinking water standard for PFOS and PFOA, two of the PFAS analogs, is in the low part per trillion range. So really, really low concentrations and of very toxic chemicals. Plastics a different scenario. I mean, the exposure to plastics all the way back to 1907, right, has just been tremendous. And so the real question is, what are the implications of that exposure? And maybe I should have said this on the slide because you saw my title, "What's in the risk?" We actually don't know yet. So I'll give you the answer first, which I think you probably know already. We're learning a lot, but we still don't have a very good understanding what the risk is. So one of the biggest issues we're having right now is the measurement of plastics, measuring, and I'm going to say micro and nanoplastics, and I'll mention this a couple of times. Obviously, the focus of the workshop or seminar here is on microplastics, but I'm going to make the argument that you can't really talk about microplastics without nanoplastics. It's all just a matter of how big the particle is. But measuring these in complex matrices, like human tissues, like soil, like food, is really, really hard. Although I am an environmental toxicologist, I run an analytical chemistry laboratory, and these are really difficult analytes to detect. And you can kind of see this from some of the controversy. So there was this paper in 2019 that came out that said we ingest a credit card worth of plastics every week. And then shortly after that, there were a whole series of papers that came out and said that's not true. It's actually far less significant than that. And that was an analytical debate. How do you measure these things? More recently, there was this paper that came out in 2025, this one down here, I'll take my highlighter, which the professor looked at the brains of cadavers and measured micro and nanoplastics and came up with a number of about 7 grams of micro and nanoplastics per brain. And somebody made the connection that 7 grams is about what a plastic spoon weighs. So then this thing just all took off. CNN's running stories about how we all have a plastic spoon in our brain by the time we die. Well, it turns out that the technique that was used was pyrolysis GCMS, and the brain is a tissue with a lot of lipids, and it turns out when you do pyrolysis GCMS with a matrix that has a lot of lipids, many of those lipids are converted to compounds that are identified as plastics. So then this paper came out, and there's been a whole series of debates since then. Estimates are about 95% of what was reported as that 7 grams actually wasn't plastics. It was a biological interference. So, you know, the point being, one of the things that we need is some more robust techniques to detect these. So I have thoughts on AI, which I won't go into, but I thought, well, let's ask AI, right? So I typed in this prompt, if you can read it, you know, what's the risk of plastic exposure and toxicity in agriculture? And I got a whole bunch of information. Most of it was really, and if you can kind of see that, that's what I put in this red box. Most of it was really focused, again, on micro and nanoplastics. So these smaller particles that, as I'll show you, you know, can create significant issues in terms of where they go and what they do. But the other part that was identified was the leaching of toxic additives, and I'll talk a little bit about that as well. But AI broke it down. I just used ChatGPT into a couple of different categories. You know, you had risks to soil health itself, impacts on the microbiome, or impacts on the physical structure of soil if the concentration's high enough. I'll show you some data in a few minutes where the genetics of some of the microbiome was changed and that altered nutrient cycling. Up top, you can think about straight toxicity to organisms. Phytotoxicity would be actual toxicity to the crop. There's a couple of mechanisms there. Co-contaminant interactions is actually where we focus a lot. So the presence of a plastic can actually increase the availability and toxicity of something else. I'll show you data for arsenic, lead, cadmium, boscalid, and PFOS. So anyway, so there was a pretty robust answer, but just out of curiosity, I asked AI when AI has been wrong in agriculture. And I got a whole bunch of examples of AI admitting it was wrong, everything from misidentifying diseased plants for healthy ones to misclassifying soils. So where are we? I think the AI may be right on this one, according to the HI, the smart people I work with. And this comes in a couple of different categories, but fundamentally, I think it's a matter of size. Over time, these materials will break down into smaller and smaller particles. The plastic polymer is, when it's synthesized, there's a whole range of additives, some of which we know, some of which are toxic elements, many of which are kind of uncharacterized organic compounds. Endocrine disruptors are among those. So in terms of how we do this, it's, if your particle is more than 5 millimeters, we call it a macroplastic. If it's less than 5 millimeters, we call it a microplastic. And if it's less than 1 micrometer, we call it a nanoplastic. This is all just semantics, if you ask me, because if you've got a plastic bag that's been sitting in the environment for 100 years or however long, in that soil where that plastic's sitting, you're going to have the whole range of particles. It's just a matter of what fraction is in the nanoscale range, which is in the micro, and what's still in the macro. And that's a moving target, right, over time. So these particles are generated by a whole range of physical and some biological processes, ultraviolet light, just physical weathering and being in soil and having organisms, burrow through them. Temperature, waste incineration is going to be a big one. I'll talk about what we're going to do on that shortly. But I think the issue here is there's a robust literature in other disciplines, including nanotoxicology, nano-enabled agriculture. We know that the smaller a particle is, the more active it is. And a lot of times that greater activity is actually greater toxicity. So we know we have a potential issue. So this was from a workshop that I was part of in 2022, late 2022 at McGill University. And it was an invited workshop. There was about 15 or 16 of us. You can see the paper there from the US, Canada, Mexico, Europe, and South America, and some people from Asia. But the idea was to really look at agriculture and try and figure out what the exposure routes were. And the motivation for that was, if you look at the scientific literature, there's actually 10 times as many papers published on aquatic systems, so rivers, lakes, streams, oceans, in terms of a plastic micro or nanoplastic causing toxicity. So there's far less known in terrestrial systems, and we got concerned about food, so we specifically focused on that. And this is just the list of exposure routes we came up with in soil, some of which we've already talked about: the mulch films, hoses, hardware. As those age, they release these particles. But also, any controlled or slow release fertilizer or pesticide that's on the market, that's a plastic coating that's mediating that. And if you're fertilizing with biosolids, that's obviously the waste stream. If you're irrigating with something other than groundwater, those are all going to be potential sources. Something that I don't think is studied enough is atmospheric deposition. These are small particles, and as a result, they can be airborne. So again, this is more on this paper, where we just kind of laid out this concept of what the risk was. And one part of the risk is just being small. And I've already said this. So whenever you see MNP, that's micro and nanoplastic. But the smaller a particle is, the more likely it is to end up in an organism, and the more likely it is to go to places in that organism where it wouldn't normally go if it were a larger particle. But the other three that we really focused on was this interaction with co-contaminants. And we got funded by USDA to look at this. I'll show you a whole bunch of data on that in a minute. But then one of the big open questions, I think, right now is plastic additives, and what those additives are, where they are going, and what they're doing. And another issue is going to be this concept of aging of plastics. When you look at the scientific literature, most of the data that's been published is exposing an organism to a pristine polystyrene or a PVC particle or something. And that's just not what we're exposed to in the environment. It's not what's in our food supply. So this, and we know this now that we've started to look at it, we being the scientific community, the older a plastic is, the more different its toxicity profile becomes. Sometimes less toxic, sometimes more toxic. So anyway, so this is this USDA grant that I was mentioning. We were funded through the USDA Food Safety and Defense Program. It was a $750,000 grant. We've got about a year left. The lead is at Rutgers. We're on it, and New Jersey Institute of Technology is on it. The focus, as I said, is almost exclusively on how plastics can influence the availability of other contaminants. So our two objectives were right here. So I mentioned this is ending in about a year. We submitted 2 additional grants last year to USDA that are currently under review, $800,000 each. So hopefully one or both of those will be funded. But the idea here is to kind of run these co-contaminant exposures and to see how the presence of one pollutant is impacting the fate and effects of another pollutant. So what are our plastics? We use polystyrene, PVC, PET. We use different sizes, micro and nano size. We use different concentrations, obviously, and we use different weathering status. And we could do the weathering thing because the Rutgers team had an artificial chamber where they could weather a plastic for the equivalent of 3 years in about a month. So we measured all the usual endpoints that you would want to measure. What's the toxicity? But most importantly, where are these contaminants going? Both the plastics and what we'll call the EPs, or the environmental pollutants, which for us was 3 toxic elements: arsenic, chromium, lead, boscalid as a model pesticide, and PFOS as a model PFOS. So I'll go through this quickly because I don't think the data is I don't want to get lost in the weeds, which is what I tend to do. So the first question you want to ask is, does the size of the plastic particle make a difference on its accumulation in plants? The answer is yes, it absolutely does. So these are the types of experiments. So we used polystyrene at 2 different sizes. We did hydroponic lettuce. We did soil-grown wheat. We looked in the edible tissue only, which is the lettuce leaves or the wheat grain. And what you see is that, the plastics and the pollutants were toxic separately, but they tended to be more toxic when they were together. That was much more evident in the lettuce hydroponic situation than it was in the soil-grown wheat, but it was there. What was really interesting was, and again, particle size is making a difference. That's the question. So here's 10 milligrams per liter polystyrene. The amount of arsenic that you found in the lettuce didn't really change whether the plastic was present or not, but it bumped up the concentration. And what you see is when that polystyrene was present, you get about twice the amount of arsenic moving into the lettuce leaves, the part that we're eating. So that was a significant finding. We published that in 2025. But the flip side also happened. So the presence of arsenic and boscalid also increased the uptake of the plastic. So we're spending a lot of time trying to figure out the mechanisms because there's a couple of reasons that could happen, which I won't go into. But I mentioned this analytical issue that we were having in the beginning. So whenever you measure a plastic in something that's a biological matrix, you need at least 2 different techniques to confirm that. You got to have one technique confirming the other. In this case, we're using fluorescence microscopy because we used some tagged plastics. They had a dye on them, but we also use pyrolysis GCMS. But the point is, here, for example, here's this data. So this is the nanopolystyrene by itself. That's the amount that's showing up in the green is the shoots. There's the roots. But when arsenic and boscalid are exposed, you have a pretty dramatic increase in the amount of polystyrene that's showing up in those tissues. But it's a function of size, because when you looked at the micro size, you didn't really see that. So again, the size is important to what's happening. So question number 2, what about the age of the plastic? As I indicated, a lot of these materials are in the environment for a long period of time. They change over time. So does the weathering status of the plastic make a difference? And it absolutely does. This is the lettuce hydroponic data again. So this is arsenic data, this is chromium, and this is lead. So shoots, roots, and total plant. If you just want to look at something like this, so this is the amount of arsenic showing up in the lettuce alone. When the plastic is aged, you have a very high level. When it's unaged, you have about half that level. So when you looked across those elements, you tended to see that same trend. The statistics always didn't hold up, but if you group the 3 metals together, it absolutely held up. So the present, the weathering status of a plastic made a big difference in terms of how much other contaminant was moving around. So this is the Rutgers part of this. So they have this model digestive system that they use. You have the oral phase, the stomach phase, the intestine, and then you've got this absorption system. They do the same types of exposures, and guess what? They got very similar data. If you had a micro or a nanoplastic exposed in the food supply, and in this case, this food was actually water. We're using more complex matrices now. But the presence of the plastic increased the accumulation across the cellular barrier of the toxic elements and the pesticides, and vice versa. So what you're seeing in an exposed wheat plant in a greenhouse in soil is the same thing that you're seeing in the human digestive system. So this has become a very active area of research for us. We got into this in late 2022, just after that workshop that we had that really triggered my interest. So I really want to just go through a couple of these other studies. I want to make sure I have time for questions, so I'm really going to go through these fast. I'll just focus on the highlights. So this was from a paper we published last year where we exposed radish in soil and hydroponic conditions to polystyrene nanoplastics, but then really wanted to look at the nutritional quality of the tuber that was produced. And the take-home message was that depending on the concentration of that nanoplastic exposure, the nutritional quality of the radish was reduced. The presence of the plastic changed the gene expression and proteomics and metabolomics of that developing plant, and those changes resulted in less nutrients in the actual tuber itself, in the food, which is a significant finding separate from any toxicity to the plant itself, because we're going to eat that and it's going to have less nutritional value for us. Another thing we want to look at is out in the environment, these crops are exposed to multiple stresses. So this was an experiment where we looked at tomato, but it was tomato that was also exposed to a fungal pathogen in soil, fusarium, which you're going to get whenever you grow tomato. And it turns out if you had exposure to the micro and nanoplastics, the level of disease pressure was more than double, 219% increase. So if that plant is stressed already and you expose it to a micro or nanoplastic, you get a kind of a compounding effect. And again, we worked on the mechanism and figured out a lot of that, but it's not really all that important for what we're talking about here. We also want to see what's just happening in soil with the soil microbiome. This is one of our studies in that case. This is a metagenomic study. So this means that you're not just looking at the organisms, but you're looking at the genes of those organisms in the soil and in the rhizosphere. And, I think you're kind of getting the theme at this point as a function of the type of plastic. So how old was it, or how weathered was it? What were its properties? How big or small was it? You had significant changes in this soil microbiome. And those changes were related to nutrient cycling, both nitrogen and phosphorus. So now we don't know how long those changes are. We don't know the dynamics of it, but it pushes the soil biochemistry away from its baseline, which is not a good thing. Foliar exposure, I mentioned this. We've done one experiment on this. Think about irrigating with contaminated wastewater, or think about plastics particles in rainfall. The exposure is going to be there. What we found here is that these particles behave just like any other nanoparticle. They accumulate on the leaf surface, they move through the stomata, they end up in the leaves, and a certain amount will move to the roots. In terms of a micro and nanoplastic, it's behaving just like any other nanoparticle that something's going to be exposed to. So you're going to get that exposure. So one of the things that we're actually doing with this USDA grant now is our Rutgers partners are creating what's called a reference library. So, we shouldn't be looking at pristine materials. We should be looking at weathered materials. And when you do that, how everybody weathers them is different, and you're going to end up with different types of particles. So the solution is create a library in one place and then make those particles available to anybody that wants to use them. So that's what this project is all about. It tends to be really expensive, but I think it's really important, because otherwise we're all working with different materials. We're going to get different results. It's going to be really difficult to make heads or tails of it. I'm going to skip that one. This was just a study where we were doing some AI stuff to see if there was enough literature there to actually start predicting toxicity. There was not. So I'll just conclude. I think we already knew this coming in. We knew the exposure was significant. What we don't know is what the implications of that exposure are. I think we're starting to get a handle on some of that now. This is just some of that co-contaminant interaction stuff. So what are we doing next? We're going to make a big move towards addressing these plastics. I don't know if you can see that price tag up there. I made an emergency capital equipment request to the state of Connecticut, your tax dollars, saying that we needed to start looking at plastic additives in the food supply. The instrument that we need to do that cost $878,000. That's 10 times more than my first house. So it just got delivered 2 days ago. So we're going to stand up that program. There's a big focus on this internationally. There's an international soil congress that's held once every 4 years. There's a huge focus on soil-borne micro and nanoplastics in that. And then this last one I'll mention, this was actually April 5th. So this was, less than 2 weeks ago. ARPA-H announced a program. ARPA is the Advanced Research Projects Agency. Think DARPA, but ARPA-H is ARPA Health, to specifically look at micro and nanoplastics in the human body and how to get rid of them. This is $144 million in the first year. And I'm part of a group at Yale and Rutgers that was invited to submit a proposal on this. So we have a meeting to discuss that tomorrow. So I will stop there because I know I went too long. My email is jason.white@ct.gov if anybody needs to talk to me and I have to run. Yeah, so I don't know why it's called plastic mulch, but it's black plastic film, or white. Sometimes it's white plastic. It's just it's used as a weed barrier. I mean, you drive past any farm in New England and you're going to see it. The discovery that it has, this is all relatively recent work. Our oldest paper was 2022. When you start looking at the literature, this has all been discovered in the last 5, 6, 7 years. There are a number of biodegradable agricultural mulches or plastics that are available. But what we've found is that sometimes those biodegradable mulches, actually, they're not biodegradable. In my mind, means mineralized. It means all the way to carbon dioxide and water. A lot of these biodegradable mulches just degrade more quickly to micro and nanoplastics. So they're not going all the way. So you really have to be careful. There are ones that are completely degradable, but they tend to be more expensive and far harder to get. So in terms of are they, they're still very widely used, and I don't see that changing anytime soon, to be honest. Yep. You know, um, I struggle as a toxicologist. I struggle about the toxicity of plastics because to a certain extent, if the plastic polymer itself was really toxic, we'd all be dead by now, right? I mean, because the exposure has been so high. So I really think it's not necessarily the plastic polymers themselves as it is the materials that are released when these things get weathered and aged in the environment. And think about a plastic bag that sits in the soil. I mean, you're not talking a couple of years until it's gone. I mean, it's literally decades. And as that's happening, all those micro nanoplastics are being produced. The smaller the particle, the more likely it breaks open and releases its additives. So I think a lot, maybe most, is chemical, but some of the toxicity we see to plants is physical. These things coat the roots and they prevent nutrients from making it into the plant. That's a physical phenomenon. So I think it's probably a combination, but mostly chemical.
You talk about airborne, sources of plastics, and I focus my purchasing on, organically grown produce and stuff. And so I mentioned, are there, I guess, two-part question. One, are there like certain coatings or things on organic reproduced crops that contain plastics? And how do those affect them? And then, of course, with the airborne exposure and other kinds of, benign exposures. Have you or anyone done like comparative analysis of plastics of organic produce? Just the whole study of a lab, going in a lab, just to like what the grocery store purchases, with organic and conventionally grown, in terms of the amount of plastics and microplastics.
So the short answer is no. Nobody has done that, and part of the answer is this analytical issue that we have. There was a group of us that were planning to put in a Superfund center. That was the call for proposals was supposed to be March 31st, but that ended up getting kicked to next year. But the proposal, and those are $10 million proposals, was to do exactly what you're talking about, to do surveillance of the food supply, to look at agricultural crops in urban versus rural settings. The state of Connecticut has a couple of municipal waste incinerators. Nobody's tracking the amount of plastic particles coming out of those stacks. The idea was to look at all that, and we'll submit it again next year. But I think the real issue is that it's a complete unknown. Nobody's really looked at it yet. And what we're going to do in this ARPA-H thing is actually develop analytical techniques. We're going to work with a couple of instrument vendors to come up with new techniques that can be used to actually detect these things at the concentrations we need to detect them at. Because I mentioned PFAS, which if we can detect it, 10 parts per trillion, most of the techniques that we're looking at for micro and nanoplastics, even the best ones, were 100,000 times above that. We're at the part per million level, and we need to get much lower. Yep.
Actually, two questions. The first is based on your comment about the toxicity of plastics and chemicals involved. I'm wondering if anyone is studying the connection between the toxicity of micro and nanoplastics in connection to the toxicity of plastic manufacturing in Cancer Alley, where cancer and other diseases are more prevalent because of those plastic manufacturing facilities nearby.
Yeah, nobody's done that yet. I think there are a lot of people thinking about that. I think there are a lot of cohort studies in different schools of public health around the country where people were looking at diseases and other causes, but all of those tissue samples still exist. So you could go back and look at those and start trying to make those types of connections epidemiologically. As far as I know, nobody's really doing that work yet. And I hate to keep, I'm an analytical chemist, right? I hate to keep coming back to the analysis, but I mean, if you can't really get below a part per million, it's hard to kind of tease apart some of those questions. But I mean, if you look at the New England Journal of Medicine and some of these other journals, there's a couple of papers a week now coming out, looking at micro and nanoplastic contamination of different organs and tissues and trying to correlate that with, different factors of morbidity, disease, things like that. So and this ARPA-H, I switched the slide. This ARPA-H push is really going to make a big difference, because it's the government putting a large amount of funding in, and I think it's going to trigger a lot of interest that already isn't there.
Okay, thank you. And my second question is, I wonder if you have any recommendations for farmers or even home gardeners and community gardeners to replace the black plastic mulch. And several years, more than a decade ago now, I was reporting on agriculture, on farming magazines, and learned about a study in Western Massachusetts that UMass Amherst was running at a local organic farm where they were using unbleached and undyed cardboard in place of plastic mulch, and found that it was incredibly effective and actually even increased earthworm activity. But do you have recommendations such as that, or other recommendations to replace that black plastic mulch?
For a community or a backyard gardener, I mean, the recommendation's fairly simple. It's more work to not use it, but if the scale's relatively small, we have a garden at my house and we don't use it, but it's, half the size of this room, so we can weed it ourselves and water it when we want to. If it's a bigger operation, there are products on the market. There's some that are cellulose based. There are some that are alginate based, where they basically look the same when you lay them down, but as the season goes, you can see them starting to degrade, and by the end of the growing season, you can just till it in because it's all carbon. It's all going to be broken down. It'll enhance microbial activity and all that. But if I had to guess, I mean, that's probably 2, 3, 4% of the market for plastic mulches, but that would probably be the best recommendation. Find a product like that, and if you can afford it, use it. Short of that, a lot of it is how effective are you at removing that plastic mulch from the field, right? I mean, 60, 70, 80 days, you're not going to see a whole lot of micro and nanoplastic production. But it's, if you can only remove 80% of what you laid and the rest is there and it's tilled in, then you got a bigger problem.
Simple question for all this trouble. I just wondered about the source for hydroponic. I mean, it's obvious what the source is in brown crops, but when you're doing hydroponic, you would think that you could keep that more sterile. Is it just the tubing?
Yeah, to a large extent, it's the hardware, also, and the agrochemicals that you're using. If you're using fertilizers, a lot of commercial fertilizers are coated to control the release or control the dissolution. Those are all plastic particles. So I mean, that's a place where I think it's easier to come up with alternative solutions. But scale-wise, what is grown hydroponically in this country versus what's grown in the field, it's a much smaller fraction.
In those small hydroponic systems that people are putting in their apartments, those are just as bad.
I don't think so. I mean, think of a PVC pipe. I mean, those first couple of years that you're going to use it, it's still going to be, it's still going to have a lot of integrity, and it's in your apartment. It's, as opposed to a large hydroponic system in a commercial greenhouse that's used year after year after year, exposed to high temperature and intense light. I mean, it's all a function of the exposure. So I wouldn't be too concerned about the system that you're describing.
Thank you very much. I'd want to know if the toxicity that comes from MNPs, does it really depend on the source of the origin of the plastic? As in, if it's petrochemical-based plastic or it's coming from plant-based like rubber plantations, do you think the toxicity will be the same, or is there any research into that?
So I think there is some research into that, but the toxicity is going to be completely different. I mean, we actually showed that PVC is different than PET than polystyrene. So the chemistry of your polymer and what's in all those additives is going to be really what drives the toxicity. And a plant-based biopolymer, probably should have very low toxicity. And the last thing I'll say is to the students in the room, we have internship programs at our place. So, students can come over, including undergraduates, graduate students. those that just need credit for a senior thesis, and those that, and some of those are paid, a lot of those are unpaid, but, you know, if you're interested, reach out, because we're always, obviously, there's a ton of work that needs to be done here, and we need smart people, and you are all smart people.
Thank you so much.